Chapter 3, Plant Kingdom. In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whittaker 1969, wherein he suggested the five kingdom classification viz. Monera, Protista, Fungi, Animalia, and Plantae. In this chapter, we will deal in detail with further classification within kingdom Plantae, popularly known as plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of Monera and Protista having cell walls have now been excluded from Plantae, though earlier classification placed them in the same kingdom. So, the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperms, and angiosperms under Plantae. Let us also look at the classification within angiosperms to understand some of the concerns that influenced the classification systems. The earlier systems of the classification used only gross superficial morphological characters such as habit, color, number, and shape of leaves, etc. They were based mainly on vegetative characters or on the endostrium structure system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial systems gave equal weightage to the vegetative and sexual characteristics. This is not acceptable since we know that very often the vegetative characters are more easily affected by the environment. As against this, natural classification systems developed, which were based on natural affinities among the organisms and considered not only the external features but also internal features like ultrastructure, anatomy, embryology, and phytochemistry. Such a classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification system based on the evolutionary relationships between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all characters and the data are then processed. In this way, each character is given equal importance, and at the same time, hundreds of characters can be considered. Cytotaxonomy, that is based on the cytological informations like chromosome number, structure, behavior, and chemotaxonomy that uses the chemical constituents of the plants to resolve confusions, are also used by taxonomists these days. 3.1 Algae. Algae are chlorophyll-bearing, simple, thyloid, autotrophic, and largely aquatic, both freshwater and marine organisms. They occur in a variety of other habitats, moist stones, soils, and wood. Some of them also occur in association with the fungi, lichen, and animals, example sloth bear. The form and the size of the algae is highly variable, ranging from colonial forms like volvulus and filamentous forms like Eulothrix and Spirogyra. A few of the marine forms such as kelps form massive plant bodies. The algae reproduce by vegetative, asexual, and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by production of different types of spores, the most common being the zoospores. They are flagellated, motile, and on germination give rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size, as in Eulothrix, or non-flagellated, non-motile, but similar in size, as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size, as in species of Iridonia, is termed as anisogamous. Fusion between one large non-motile static female gamete and a smaller motile male gamete is termed oogamous. Example, volvox and fucus. Algae are useful to man in variety of ways. At least a half of the total carbon dioxide fixation on the earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producer of energy-rich compounds which form the basis of food cycles of all aquatic animals. Many species of Porphyra, Laminaria, and Sargassum are among the 70 species of marine algae used as food. Certain marine brown and red algae produce large amounts of hydrocolloids, water holding substances, example algin, brown algae, and carrageen, red algae, which are used commercially. Agar, one of the commercial products obtained by Gelidium and Gracilaria, are used to grow microbes and in preparations of ice creams and jellies. Cholera, a unicellular algae rich in proteins, is used as food supplement even by space travelers. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae, and Rhodophyceae. 3.1.1 Chlorophyceae. The member of Chlorophyceae are commonly called green algae. The plant body may be unicellular, collinear, or filamentous. They are usually grass green due to the dominance of pigments chlorophyll A and B. The pigments are localized in definite chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made of an inner layer of cellulose and outer layer of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated zoospores produced in zoosporangia. The sexual reproduction shows considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamous, or oogamous. Some commonly found green algae are Clydomonas, Volvox, Elothrix, Pyrogera, and Chara also we could say, no? 3.1.2 Phaeophyceae. The members of Phaeophyceae or brown algae are found primarily in marine habitats. They show great variation in size and form. They range from simple branched filamentous form, ectocarpus, to profusely branch formed as represented by kelps, which may reach a height of 100 meters. They possess chlorophyll A, C carotenoids, and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophylл pigment fucosanthin present in them. Food is stored as complex carbohydrates, which may be in the form of laminarian or mannitol. The vegetative cells have a cellulosic walls, usually covered on the outside by gelatinous coating of algin. The protoplast contains, in addition to plastid, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a holdfast and has a stalk, the stipe, and the leaf-like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in most brown algae is by biflagellate zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous or anisogamous or oogamous. Union of gametes may take place in water or within the Oogonium species. The gametes are pyriform, pear-shaped, and bear two laterally attached flagella. The common forms are Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. Figure 3.1b. 3.1.3 Rhodophyceae. The members of Rhodophyceae are commonly called red algae because of the predominance of the red pigment R phycoerythrin in their body. Majority of the red algae are marine with the greater concentrations found in the warmer areas. They occur in both well-lightened regions close to the surface of the water and also at the great depths in the ocean where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization developments. The common members are Polysophonia, Porphyra, Gracilaria, and Gelidium. 3.2 Bryophytes. Bryophytes includes the various mosses and liverworts that are found commonly growing in moist, shaded areas in hills. Bryophytes are also called amphibians of the plant kingdom because of these plants can live in soil but are dependent on the water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in plant succession on bare rocks soil. The plant body of bryophytes is more differentiated than that of the algae. It is thallus-like in prostate or erect and attached to the substratum by unicellular or multicellular rhizoids. They lack true root, stem, or leaves. They may possess root-like, leaf-like, or stem-like structures. The main plant body of the bryophytes is haploid. It produces gametes, hence is called gametophyte. The sex organs in bryophytes are multicellular. The male sex organ is called antheridium. They produce biflagellate antherizoids. The female sex organ called archegonium is flask-shaped and produces a single egg. The antherizoids are released into water when they come in contact with archegonium, and antherizoids fuses with the egg to produce the zygote. Zygote do not undergo reduction division immediately. They produce a multicellular body called sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division meiosis to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous mammals, birds, and other animals. Species of sphagnum, a moss, provide peat that have long been used as fuel and as packing material for transshipment of living material. Because of the capacity to hold water, mosses along with lichens are the first organisms to colonize rocks and hence are of great economical importance. They decompose rocks, making the substrate suitable for the growth of higher plants. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. Liverworts 3.2.1. The liverworts grow usually in moist, shady habitats such as bank of streams, marshy ground, damp soil, bark of trees, and deep in the woods. The plant body of a liverwort is thyroid, example Markensia. The thallus is dorsoventricle and closely pressed to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. Asexual reproduction in liverworts take place by fragmentation of thalli or by the formation of specialized structures called gammae, singular gamma. Gamma are a green multicellular asexual buds which develop in a small receptacles called gamma cups located on the thalli. The gamma becomes detached from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organs are produced either on the same or on different thalli. The sporophyte is differentiated into foot, seta, and capsules. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophytes. 3.2.2 Mosses. The predominant stage of life cycle of a moss is the gametophyte, which consists of two stages. The first stage is the protonema stage, which directly form a spore. It is creeping, green, branched, and frequently filamentous stage. The second stage is the leafy stage, which develop from the secondary protonema as a lateral bud. They consist of upright, slender, excess-bearing, spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoid. This stage bears the sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organ antheridia and archegonia are produced at the apex of the leafy shoots. After fertilization, the zygote develops into sporophyte consisting of a foot, seta, and capsule. The sporophyte in the mosses is more elaborate than that in liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanisms of spore dispersal. Common example of mosses are Funaria, Polytrichum, and Sphagnum. 3.3 Pteridophytes. The pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plants to possess vascular tissues. Xylem and phloem. You shall study more about these tissues in chapter number 3. The pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil conditions. You may recall that in bryophytes that the dominant phase in the life cycle is the gametophytic plant body. However, in the pteridophytes, the main plant body is sporophyte, which is differentiated into true root, stem, and leaves. Figure 3.3. These organs possess well-differentiated vascular tissues. The leaves in pteridophyte are small macrophytes, as in Selaginella, or large macrophytes as in ferns. The sporophytes bear sporangia that are subtended by a leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structures called strobili or cones Selaginella equisetum. The sporangia produce spores by meiosis in spore mother cells. The spore germinate to give rise to inconspicuous, small but multicellular, free-living, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, shady places to grow. Because of these specific restricted requirements and the need for the water for fertilization, the spread of the living pteridophytes is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organ called antheridia and archegonia, respectively. Water is required for transfer of entherozoids. The male gametes released from the antheridia to the mouth of the archegonium. Fusion of male gamete with the egg present in the archegonium results in the formation of zygote. Zygote thereafter produces a multicellular, well-differentiated sporophyte, which is the dominant phase of pteridophytes. In majority of the pteridophytes, all the spores are of similar kinds, such as plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macrolarge and microsmall. Spores are known as heterospores. The megaspores and the microspore germinate and give rise to female and male gametophytes respectively. The female gametophytes in these plants are retained on the parent sporophytes for variable periods. The development of the zygotes into young embryos take place within the female gametophytes. This event is a precursor to the seed habit, considered as an important step in evolution. The pteridophytes are further classified into four classes: Psilopsida, Psilocytum, Lycopsida. Examples are Selaginella, Lycopodium, Spinospida, Equisetum, and Pterospida, Diopteris, Pteris, and Adiantum. 3.4 Gymnosperms. The gymnosperms, gymnos, naked sperma seeds are plants in which the ovule are not enclosed by any ovary wall and remain exposed, both before and after fertilization. The seeds that develop post-fertilization are not covered, that is, are naked. Gymnosperms include medium-sized trees or tall trees and shrubs. One of the gymnosperms, the giant reedwood tree squaw, is one of the tallest tree species. The roots are generally tap roots. Roots in some genera have fungal association in the form of mycorrhiza, pinus, while in some other cases, cycas, for example, small specialized roots called caloroid roots are associated with nitrogen-fixing cyanobacteria. These stems are unbranched cycas or branched pinus citrus. The leaves may be simple or compound. In cycas, the pinnate leaves persist for a few years. The leaves in the gymnosperms are well adapted to withstand extreme of temperature, humidity, and wind. In conifers, the needle-like leaf reduce the surface area. Their thick cuticle and sunken stomata are also help to reduce water loss. The gymnosperms are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne on sporophylls, which are arranged spirally along an axis to form lax or compact strobili or cones. The strobila bearing microphyls and microsporangia are called microsporangiate or male strobili. The microspores develop into male gametophytic generation, which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called a pollen grain. The development of the pollen grain takes place within the microsporangia. The cones bearing megasporophylls with ovules or megasporangia are called microsporangiate or female strobili. The male or the female cones or strobili may be borne on same tree, that is pinus. However, in cycas, male cones in megasporophylls are borne on different trees. The megaspore mother cell is differentiated from one of the cells of the nucleus. The nucleus is protected by envelopes and the composite structure is called an ovule. The ovules are borne on megasporophylls which may be clustered to form the female cones. The megaspores mother cell divide meiotically to form four megaspores. One of the megaspores enclosed within the megasporangium develops into multicellular female gametophyte that bears two or more archegonia or female sex organs. The multicellular female gametophyte is also retained within the megasporangium. Unlike bryophytes and pteridophytes, in gymnosperms, the male and female gametophytes do not have an independent free living existence. They remain within the sporangia retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules borne on megasporophiles. The pollen tube carrying the male gametes grow towards archegonia in the ovules and discharge their contents near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and ovules into seed. These seeds are not covered. Angiosperms. Unlike the gymnosperms where the ovules are naked, in the angiosperms of flowering plants, the pollen grains and ovules are developed in specialized structure called flowers. In angiosperms, the seeds are enclosed in fruits. The angiosperms are an exceptionally large group of plants occurring in wide range of habitats. They range in size from the smallest Wolfia to tall tree of eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicines, and several other commercially important products. They are divided into two classes, the dicotyledons and monocotyledons, figure 3.5. The dicotyledons are characterized by seeds having two cotyledons, reticulate venations in the leaves, and tetramerous or pentamerous flowers, that is, having 4 or 5 members in each whorl. The monocotyledons, on the other hand, are characterized by single cotyledonous seeds, palevenations in the leaves, and trimerous flowers having 3 members in each floral whorls. The male sex organ in the flower is the stamen. Each stamen consists of a slender filament with an anther at the tip. Within the anthers, the pollen mother cells divide by meiosis to produce microspores which mature into pollen grains. The female sex organs in a flower is the pistil. The pistil consists of a swollen ovary at its base, a long slender style, and a stigma. Inside the ovary, ovules are present. Generally, each ovule has a megaspore, mother cells, that undergoes meiosis to form 4 haploid megaspores. 3 of them degenerate and 1 divide to form the embryo sac. Each embryo sac has a 3-celled egg apparatus, 1 egg cell, and 2 synergids, 3 antipodal cells, and 2 pollen nuclei. The pollen nuclei eventually fuse to produce a diploid secondary nucleus. The pollen grain after dispersal from the anthers are carried by wind or various other agencies to the stigma of a pistil. This is termed as the pollination. The pollen grain germinate on the stigma and resulting pollen tube grows through the tissues of the stigma and style and reach the ovule. The pollen tubes enter the embryo sac where 2 male gametes are discharged. One of the male gametes fuses with the egg cells in gamete to form a zygote. The other male gametes fuses with the diploid secondary nucleus to produce the triploid primary endosperm nucleus pen. Because of the occurrence of the two fusions, that is syngamy and triple fusion, the event is termed as a double fertilization, an event unique to angiosperms. The zygote develops into an embryo with 1 or 2 cotyledons, and the pen develops into endosperms, which provide nourishment to the developing embryo. The synergids and the antipodal degenerate after the fertilization. During these events, the ovule develop into seeds and ovaries develop into fruit. The life cycle of the angiosperm is shown in the figure below. Plant life cycles and alteration of generation. In plants, both haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies, haploid and diploid. The haploid plant body produces gametes by mitosis. This plant body represents a gametophyte. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. This in turn divide by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of generations between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals representing them differ in the following patterns. First is sporophytic generation is represented only by the one-celled zygote. There are no free-living sporophytes. Meiosis in the zygote results in the formation of haploid spores. The haploid spores divide mitotically and form the gametophyte. The dominant photosynthetic phase in such plant is the free-living gametophyte. This kind of life cycle is termed as haplontic. Many algae such as Volvox, Pyrogyra, and some species of Clydomonas represent this pattern. Second, on the other extreme is the type wherein the diploid sporophyte is the dominant photosynthetic independent phase of the plant. The gametophytic phase is represented by the single to few celled haploid gametophyte. This kind of life cycle is termed as diplontic, and algae fucus species represents this pattern. In addition, all seed-bearing plants, that is gymnosperms and angiosperms, follow this pattern with some variation wherein the gametophytic phase is few to multicelled. Bryophytes and pteridophytes, interestingly, exhibit an intermediate condition, haplodiplontic. Both phases are multicellular, however, they differ in dominant phase. A dominant independent photosynthetic thalloid or erect phase is represented by a haploid gametophyte, and it alters with a short-lived multicellular sporophyte totally or partially dependent on the gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. The diploid sporophyte is represented by a dominant independent photosynthetic vascular plant body. It alters with a multicellular saprophytic autotrophic independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplontic life cycle. All pteridophytes exhibit this pattern. Interestingly, while most algal genera are haplontic, some of them are, such as Ectocarpus, Polysaphonia, kelps are haplodiplontic. Fucus and alga is diplontic.